So welcome to today's webinar or presentation. I am Kylie, the Regional Field Application Scientist of Azure Biosystems. All right. So in case you haven't heard about Azure Biosystems before, this is a United States brand, okay? And the equipment we are talking today is a next generation fluorescence laser-based scanning system. I know it's lengthy, but this is our most proud and highest spec equipment in our company portfolio. And you can see we launched it late 2023, and then we have a lot of installations in these years. Okay, so this will be the equipment we're talking about today and how the researcher in microbiota or microbiome field utilize it for their research or R&D. Okay, so let you get a glimpse of how a zoo image or our protein expressions and all kinds of visualizations. So we have three main solution to image your result or to visualize your result. So we have a zoo imaging systems, we have Chemisolo, and we have Sapphire FL. Okay, so overall, these are the application overview that can be covered by a cumulative of these three equipment. Okay, so how we separate them in terms of their roles is in a zoo imaging system is your powerhouse, okay, in the laboratory for your basic nucleic acid gels or colorimetric gels, okay, or anything up to chemiluminescence signal and also fluorescence signals on your blood or on your gels. Okay. And this is the very generic equipment that you can utilize in your laboratory. Okay. And we have something more niche called a chemisolo, which is dedicated for image to image chemiluminescence signals only. Okay. So where is Sapphire FL? So this is when Sapphire FL Biomolecular Imager comes in. Okay, this is when you want or we want to look at a fluorescence signals. Okay, but we are not only looking it in the blood or on the gel, we're looking it in a way of we want it to be high resolutions and very good signal to noise ratio and clean background. Okay, and this is when Sapphire FL will be coming in. Okay, so we're not only using Sapphire FL for these blood and gel, it can be used on image to image your array, okay, and your plate base, okay, either be 96 well, 6 well plate is up to you, okay, because they are all similar size, okay, and of course, in this 2013 version of Sapphire FL or our latest Sapphire, okay, we can use it to image live small animal, okay, and of course, not forget you can also use it to do your phosphor imaging on radio labeled samples. Okay, so you might be wondering how we come so far, okay, in terms of fluorescence or advanced high resolution imaging. Okay, so let's see, you have fluorescence samples regardless in your research, okay, all right, and then you have the samples existed in membranes, in gels, either they have been fluorescently labeled or you tag them with fluorophores. Okay, and this is when you can look at the differentiations in terms of high resolution imaging. Okay, bring it forward. You can also image it with multi-wells, plate, 96, 12, up to you. Okay, and moving further, we can look into slides, glass slides this time, okay? Or we can say optical grade bottom slides, okay? So it can be applied for your microarrays and tissue sections. And of course, biological samples, okay, like your organ, plant, small animals, and agar plate as well. Okay, so these are the main applications, or I would say the samples that you can use in Sapphire FL biomolecular imager. But what is Sapphire FL? So it is the only multimodal laser scanner on the market right now, okay? Where we have the FL stands for two words. One is a fluorescence, and the second word is flexibility, okay? Right, so fluorescence means in this equipment, you can interchange your laser and filter, and then in that sense, you can mix and match your laser and filter combination, okay? We'll we'll dig deeper into these sections later. Okay. Right. So how about the flexibility in Sapphire FL? So the flexibility here is we're talking about a very wide dynamic range. We're talking about 24 bit. Okay. And then you have adjustable focal plane, okay, with 0.01 mm increment, okay, high resolutions for you to choose from, large field of view, 25 cm square, and you'll be able to image living small animals, okay, and also chemiluminescence module and phosphor imaging for you. Okay. So this is the fire alpha in a summary. So let's look at how we use it. Okay. So firstly, look inside the technology. It has these user interchangeable laser and filter modules. Of course, it comes with a standard set, okay, like your 488, Psi 2, Psi 3, Psi 5, near infrared 700, 800, okay, and the phosphor imaging, all right. So these will have their own as head of laser and filters. So the filters here you can see in the middle photo, okay, it exists in the form like a USB drive where you can freely switch and match, okay, your emission filter with any laser if you have them. Okay. So what's the benefit of this matching laser or mix and match lasers and filters? Firstly, okay, the first benefit, the much more obvious one, which only having it in the Sapphire FL right now, is this ADAPI module, optical module. Okay, so here's the in-cell Westons where we stained the HeLa cells with a DAPI. So we know DAPI is a very generic nucleic acid stained. Okay, so it's for any nucleic acid, okay, or nucleus. Okay, right. So if we scan it or image it under 488 channel or your Cyto channel, Cyto channels, you will see that there is no signal that you can visualize. But however, if you image it under our DAPI modules, you're able to see the signal very clearly according to the serially diluted cell count. Okay, so that's a benefit. You'll be able to visualize them at maximum performance. So this is an uncoupled modular design. Uncoupled means the laser and filter they are not bound together. You can mix and match them. So in a way, it was always ready to meet your needs. So here's a list of our standard optical module, which you can see we paired it with the laser and filter. For example, this is for your DAPI, our standard 488, okay, and side 2, side 3, side 5, and near infrared. Okay. So since this is a modular design, first you can maximize the sensitivity. One example is the yellow fluorescence protein here. You can see it has a different emission wavelength or spectra than our standard Psi2 channels. Okay, so with that, we can ensure you'll have a maximum data collection and optimize, okay, excitation and emission wavelength for the detections and visualizations. And for you as a user, or for us as a user, we only need to choose the modules that we need and expanding. Okay, let's say you have a signal inside two channels, and one day you have a classmate that has YFV or YFP, sorry, yellow fluorescent protein uh, reporters. And what you need to do is only having extra filters for that. Okay, and it's very easy to change. You can basically change it as your own. It's been optimized. The equipment has been developed in that way. The user can be easily change the filters and also the whole modules. Okay, so the whole excitation emission wavelength, we are talking about fluorescence channels here. Okay, it covers from our UV range up until near infrared range, as you can see from here. Okay, so and of course, if you are looking for a very custom or you are developing a new dye or looking for a certain reporter or marker that has a different kind of excitation and emission wavelength, they are not bound to the standard, let's say, Sci 2, Sci 3, Sci 5 channels. Okay, and you can't really find it on the market. And Sapphire FL will be the perfect equipment for you because we can customize the excitation and emission module for you. Okay, so to match the excitation emission to your needs. One of the example is the TD tomato. Okay, as you can see at the standard side 3 channels, okay, where 562 excitations, okay, we can see that it is not falls at the, let me see how many percentage here. This is roughly around maybe 60%, okay, excitation efficiencies, okay, and also around 60% of your emission efficiencies, okay. But in Sapphire FL, we have can allows you to do custom lasers, okay, right? So you can see where we can hit the exact, the best efficiency for your excitation emission. Translate to your research will be the best signal to noise ratio, the best visualization and data sensitivity that you get in your research. Okay, so all these fluorescent signals are available in the extended dynamic range feature. Okay, so this is what I mean when it has a 24 bit, okay, signals, a wide signal linearity available for all the fluorescent signals. Okay, so extending your signal linearity in your research or in your samples with EDR, extended dynamic range. So the difference here is with the EDR, you'll be able to generate a single image, okay, which your highest express samples are not saturated, which means you will be able to quantify the or analyze the data from the image right now. Okay, instead of you have to do a lot of tinkering, okay, on let's say your knockout or knockdown samples, wild type, and your overexpressed samples. Okay, so here's how we able to achieve in the sense of 24-bit dynamic range. So these equipments via FL is not only for your like generic membrane, okay, or gels. It also covers anything up to plus 6 mm depth. Okay. So we have from -1 plus 6 mm depth, okay, and this is the focus plane or Z plane adjustable for different samples and with different depth. Okay. All these available with a click of a button. And then extending from this feature, you'll be able to do an automatic Z scan, okay, at different depths that you set, like per increment, okay, and then you'll be able to generate images like this, okay, different axis images with different expressions of the samples. Because these okay, this feature will be much more applicable to samples with depth, with thickness. Okay, for microbiology, microbiome or microbiota research, this maybe will be not that relevant, but nonetheless, this is something that you can use if you extending your research into, let's say, a gut, okay, or organ-like visualization. Okay, so of course, with adjustable z-axis, it comes with a wide resolution for you to choose from. In Sapphire FL, we'll be able to choose from 1,000 micron, okay, imaging resolution up to 5 micron resolutions. The 5 micron resolution will be something we're looking at very tiny details, such as your microarray or something like your tissue section slide. Okay. And it also has a built-in gaseous anesthesia port that compatible with your third-party anesthesia systems. So Sapphire FL supports in vivo, okay, to sorry, to in vivo studies. Okay, it basically anything that has a very good signal, like your fluorescence signals, and very good penetrations, and without much background noises. We're talking about autofluorescence signals from your equipment, but your plastic wares or the for the fur or organs or some sort. This is what Sapphire FL will be excel in. So it will be able to give you that best signal to noise ratio. So in that sense, the Fire FL has endless application possibilities. Okay, as you can see here, these are the samples that you can be used or is being used in a Sapphire biomolecular imager. Okay, membrane, gels, radioactive samples. Okay, under the umbrella of fluorescent and radioactive samples, and then you can image your membrane, gel, slide, multi-well plates, and all the others listed over here. So let's look at these applications, or we look at the performance of this equipment, Sapphire FL, okay, and also look at the publications that in microbiota and microbiome research. So firstly, here's the list of the standard optical module that has the laser and filter pad ready for you to image your your fluorophore, your labels, or your reporters. Okay, and here you can see there are two types of detectors. Okay, that we actually put for this module. So each of these TMT and APD, they are the best detector on the market in terms of fluorescent signals and also partly and radioisotope or phosphor imaging. Okay, so here's the pairing that is dedicated. We pair it for the best quantum efficiency for your signal visualizations. Okay, so first example we're looking at will be phosphor imaging. It is used to image your radioactive samples. It has a higher sensitivity because it requires a labeling and also a phosphor screen to keep the signal. So assays that require radioactivity, okay, and you can use or reuse the phosphor screen many times. It's repeated for many experiments. So your sample could be one off, but the screen can be stored and is a repeated use. So, what are the radioactive samples or the dye or the things that we use? So, the very generic one will be the hydrogen free, okay, carbon 14, phosphorus 32, this is phosphorus, yeah, 32. So, these are the tag or radio label for your protein, lipid, glycoprotein, polysaccharide, or nucleic acid. Okay. And we have also something more to the nuclear side of the radioactive, like iodine 125 and so on. Let's look at the sample right now. So this is a carbon 14C labeled brain section. As you can see, here's the details where you have the brain sections exposed to image plate or phosphor screen for 3 days and then scanned at 25 micron resolutions. Okay, here's some brain section, tissue sections, and next, looking here are the signals from the nuclear radioisotope, iodine 125, actinium 225, and so on. In terms of biome or microbiota research, we're looking at here is a very new publication from eLife. Okay, here's I am to be just a disclaimer, I'm not a microbiologist. So I just what I do here is I try to summarize what's the research do, okay, what's the publication trying to do or use Sapphire or Sapphire FL in the research. Okay. So the first publication we're looking at here is from eLife with the title Core Variations Analysis Reveals Elements of Selectivity in quorum sensing systems. Okay, so we are talking here is a bacterial community or communication, which is a cell-cell signaling not really sort of pathway, but it's kind of a mechanism called acrylyl homoserine lactone AHL quorum sensing. So the research trying to understand how the protein in this signaling called evolve. The technique they use here is basically a thin layer chromatography screening on the proteins that use in acryl homoserine lactone. Okay, so what they do is actually they extract the carbon 14C labeled meteor 9 feed it to the bacteria. Okay, and then the TLC plate exposed to phosphor screen and image with Sapphire Biomolecular Imager. On the right over here, here is the example in the supplementary figure. Okay, here's a thin layer chromatography analysis of the carbon 14C labeled AHLs extracted from the bacteria with the indicated amino acid substitution. So here is one of the techniques that they use to identify or to extract or purify the AHLs. Okay, so here's an example used in phosphor imaging, radioisotope or radio labeled samples. One of them is in TLC. Next will be something more generic to us, a fluorescence Western blot to detect your protein expressions, okay, in your cell lysate or so on and so forth. So in fluorescence Western blot here, you can see clearly they are mostly in either a single target or multiple target, or we call multiplex Western blot over here, okay, where you can detect multiple protein targets on the same blot. Okay, so here's some example on the image. Okay, right. So in microbiome or microbiota research, we have a publication from PNAS. I think this is from this year as well. Okay, so they also utilize these fluorescent uh, Western blot technique, okay, to look at or validate the deletion in the mutant group. Okay, so here's more to confirmation of the research. Oh, no, sorry, the sample group that they do knockout, deletions, okay, or the wild type. All right. So this is a very generic technique. Okay, and then of course they use why they have to use fluorescence Western blot because it since we can use it on like a zoo imaging system, the normal gel documentation system that we can use on the laboratory, because here in Sapphire FL, it gives you the better signal to noise ratio and a wider dynamic range than your normal CCD or CMOS camera, and a best signal to noise ratio, clean background, okay, and of course the best sensitivity as well compared to CCD and scientific CMOS. Okay, so this research is what they do is they are able to identify a new family of LDTs, okay, LD transpeptidase that is distinct or different than previously known LDTs. So they establish a new class of bacterial peptidoglycan cross-linking enzyme, and then they tested it and see whether it's functional, okay, or with the deletion, and then to prove that it's functional, and suggest it for select as a target for selective antibiotic. Okay, so this is not really restricted within a microbiome or microbiology research, but it's more about covering on the targeted antibiotic or drug discovery here. Okay, but more about fundamental. A next example will be also utilizing a fluorescence Western blot, which is a publication from Gut Microbe. Okay, so we're looking at rodentum over here. So these researchers trying to explore the role of type 2 secretion systems or T2SS during the natural host infections. So what they use is basically a protein secretion assay, and then they immunoblotting with fluorescent labels or fluorophores, okay, on on the secretes or extracts, and to determine the T2SS, the presence of them, and also the secreted protein effector. And also they do functional tests on the knockout or the knockout or knockdown that or T2SS in the bacteria will lead to reduced systemic spread infection and also defective colonization during the host infection. So this data, representative data that we extract from the paper. Okay, so this is a representative image from their fluorescence immunoblot. All right, and then also another one fluorescence spectrum blot technique used in microbiome research. Okay, here will be a very simple looking at the here where is that kid? Yeah, validation of the knockout using the fluorescent Western blot techniques. Okay, right. And then there's a lot more. Okay, research in microbiome and microbiota research. They utilize Sapphire Biomolecular Imager on the techniques using the techniques of fluorescence microscope. Okay, as I mentioned before, why they want to use this equipment because it gives you the best signal to noise ratio, better, I'll say the better signal to noise ratio, and a wider dynamic range compared to our CCD and scientific CMOS camera. And of course, let's not forget about in-gel fluorescence. Now we are moving on to gel. Okay, fluorescently labeled gels. Okay, here the fluorescence signals can be coming from your samples. Either you tag the samples, okay, either tagging a protein or you already have a plasmid that has that reporter of, let's say, GFP or so and so forth. Okay, let's look at the first publication that utilizing the fluorescence gel. Okay, so here's a publication from Nature Chemical Biology. Okay, so they're studying uh hang on. So they're studying the inhibitors of the colibactin biosynthesis, okay, which is a very uh, strong factor in gut bacteria genotoxin in the colorectal cancer development. So this is more about a gut environment then. All right. So the assay they use is basically called activity-based protein profiling, BPP, of the penicillin binding proteins, sorry, penicillin binding proteins. So there are, of course, some prerequisites steps they need to do before they come to this step where they load into a gel, okay, and fluorescently label them, okay, and we're able to look for the possible targets of their inhibitors, okay. So here's one of the representative image they can find in their supplementary data. And next, we're looking into 2D diet. Okay, so it's a publication that study micro effects on a breast cancer cell lines. So this is example of a 2D diet, okay, on the page gel, okay, that we can look at the proteomic changes affected by the bacteria itself. So extending from the gel or fluorescently labeled gels, Sapphire FL also can image your Comessi blue stained gels, which at the 685 optical module, or we call near infrared 700 channels. Okay. So we are done with the gel. Let's move on with a cell-based assay, which may be much more relevant to microbiome or microbiota research. Okay. So first we have here, which is a PBMC stain with AO PI, the scan with Sapphire FL, okay, where we can look at the live and dead cells assay on cell counting slides, okay, and then we also can look at scan a plate. This is a 24 well plate, scan at 100 micron resolution within 7 minutes to look at the yellow fluorescent protein expressions, okay, and this is more about testing a different transfection kit uh, that efficiency on the samples. Okay, so here you can see very obviously the first transfection kit has the best transfection efficiencies compared to the rest of the two. Okay, so here's I'm saying here not only the Sapphire FL able to scan or visualize your fluorescently labeled samples, we're also able to scan it at a very high speed. So in a cell-based assay, a 96 well plate or a 24 well plate, since they are at the same size, okay? So 100 micron resolution or pixel size scanning will take you only 7 minutes plus minus. Okay, right. So extending from there, we can also image cyan fluorescent protein or yellow fluorescent proteins in the cells. So cyan fluorescent protein is a type of marker right now, which is being used, okay, to tag on the DNA. Okay, so you can see here the excitation is actually falls at the laser excitation falls around 450, okay, and the emission is actually around 470 or 480 plus. Okay. So on Sapphire FL, we have an optical module dedicated for these label or these fluorophores, cyan fluorescent protein. Okay, you can see here this is belongs to our standard optical module. Okay. So this is also explaining how a Sapphire FL able to image a wide variety of dyes and reporters that you have in your samples. Right. So extending from cell-based assays, we can look at it at the microplate format. Okay. This is a very useful technique or visualization style. Okay. Where you can do a high throughput either protein detection or signal or changes in terms of a certain metabolite. This is something that you can see, okay, on the image and analyze from it. So what people use it for will be mostly not like covered around these regions and also extended from it will be protein phosphorylation in signaling, compound screening, okay, compound screening on your microbe, okay, or RNA or nucleic acid library screening, codification of viral load, or you can screen your monoclonal antibody clones or do some cell proliferation studies characterize your functional characterization on your mutant receptors, yes or no, or how much, okay? And you can also look at like cell receptor trafficking or internalization, or you can check the silencing knockdown of the cell surface protein as well at a very high throughput manners. Okay, right. So let's look at the publication that use microplate cell-based visualization or assay, okay, in the microbiota or microbiome research. So the first publication here will be from Archive of Toxicology, okay. So the title is Urolithin A, attenuate arsenic-induced gut barrier dysfunctions. So this research is mainly to study the benefits of gut microbiome metabolite, which is urolithin A, against an inorganic arsenic-induced abnormalities on the gut epithelium. Okay, so here they actually use the Sapphire for two plate-based assay. First is they're detecting the mitochondria membrane potential. Okay, and second thing they examine will be on the ROS generation. So they're looking at the changes in fluorescence signals in 96-well format. Okay, so as I mentioned, because everything can be scanned at 100 micron resolution within 7 minutes, this is actually at a very quick and fast speed able to generate the result. A very direct comparison between your experimental groups. Okay, so extending from these plate-based assay or microplate assays, we have something called in-cell westerns. Okay, so in-cell westerns, like our Western blot, we're talking about immuno tagging here. Okay, so we're talking about immuno, but not on the blot, but in the plate. Okay, so we also have one publications. Okay, where the group of researchers study the trichotillins, okay, together with the genotoxins from the intestinal microbiota and pesticides. So in-cell Western they use here, okay, uh, technique is used in two measurements. One is ribotoxicity, and second, they are looking at the DNA damage by calculating the genotoxic index. Okay, so these are representative image in a graph format to look at the data at those depending manners. Okay, so in-cell Western, just a uh, brief about the in-cell Westerns itself. Okay, it's a very high throughput and accurate measurement to measure your intracellular or extracellular protein expression in situ. Okay, but Sapphire FL is not a microscope. Okay, so what we are looking at here is a general fluorescence signals across well to well. Okay, so from here you can see it's immunotagging. So we have these two housekeeping GAPDH, RNAP kinase here. Okay, and then we have this normalization control using a total cell state. Okay, and so we have these in a multiplex manner, so it can facilitate your data uh, collections. With that, we have a kit called a Cyto Easier Western kit. So if you're interested in exploring this kind of application, we have a optimized kit for you to use that covers all the reagent you need in to perform the in-cell Westerns. Of course, we don't have your samples and your primary antibody. Okay, right. Next, we're looking into a higher resolutions right now, tissue array. Okay, in matched tumor or normal tissues in microarray scan at 10 micron resolutions. The bear in mind that each of these tissue samples is about 1.5 mm in diameter, so they are very small. And next thing will be something here in near infrared microarrays. They're looking at cytokines expression in different spots. Okay, these require high resolution imaging, like 10 or 5 micron resolutions. Here's an example of a tissue section slide scanned at 5 micron resolutions. Okay, as I mentioned, it is Sapphire FL is not a microscope itself, but it is a very good, I'll say super good in taking high resolution images up to 5 micron resolutions. Okay, right. So in normal circumstances, you may be replacing the zoom in image with your confocal microscope images. Of course, Sapphire FL, because it has this built-in gaseous anesthesia port, allowing researchers or us to image live animal. Okay, while it is not, it can capture a very publishable, it can capture publishable images of a fluorescent tumor in vivo. So here we have mice that injected with red fluorescent protein expressing 41 cell and image in 100 micron resolutions. So we can see the shape and calculating the size of the tumor in vivo. And of course, validations of the tumor size ex vivo also can be done. And here's the Z scans we showed in the GIF format to show a different expressions or the sizes or the shape of the tumor cell, tumor in harvest from the mice. Okay. So here will be some. These are the very general, I would say, samples that we can see and use in Sapphire FL. But nonetheless, we can also use it to image like living zebrafish. So, so this is the demo data that we captured. Okay, so we can see there's the Alexa Flow 488. Okay, and also near infrared 800. Okay, labeled in their, I think this is abdominal regions of the zebrafish, and this is scanned at 5 micro resolution with focus setting at 2 mm. Okay, because we can adjust the focus, so we can determine the best focus for each of your sample with different depths. Okay, so here's a study distribution of exosomes. Okay, where they do label the lipophilic exosome that inject into the zebrafish. So these can be used to track the distribution and possible penetration through the blood-brain barrier. So that's their motto, the aim of this research. Kiwi, fruit, all the living organism, they have autofluorescence signals. So we utilize autofluorescence signals from the fruit itself. So the cross sections of the kiwi, we can see here, scan at 10 micro resolutions. Fungus plate, will be able to scan fungus plate, okay, at 100 micro resolutions. You can see here at different channels representing a different markers or different metabolites in the fungus. Leaves, okay, so we have leaves scan at side 2, side 3, and also side 5, not say side 5 channels, where the chlorophyll is actually at side 3 channels, is autofluorescence signals. We can compare the leaf color as an indicator for change, let's say in the metabolite inside the leaf, or the difference in the chlorophyll content, okay, but affected by your, let's say, pound screen for leaves and so forth. Another example of leaves, which is a tobacco leaf, okay, scan at for a GFP signal at Site2 channels at 20 micron resolutions. Okay, so this is more about infections, disease, resistance, okay, where they want to develop resistance in the tobacco leaves. Mosquito, okay, we can also image a mosquito that fed with your 488 labeled sugar water. You can see here under 488 channels, you can look at it's actually covering the entire abdominal area of the mosquito, but we don't really see it in the exoskeleton of the mosquitoes. And finally, we have this very proud image that we have scanned the honey lemon on Sapphire Biomolecular Imager and won the WikiScience competition back in 2019. So this signal is basically autofluorescence. Here's some selected publications. Okay, Sapphire FL has been used widely and extensively for your general like fluorescent plots, gels, and anything up to very high resolutions, uh, like microarrays and so forth. So here's some examples of publications that you can actually found. We have the database in our on our website that you can search through the publication and look at how people utilize the Sapphire in their research. We also have it in the publication booklet format, okay, to showcase the performance and how the researcher use it. Different samples you use on the Sapphire biomolecular images. Here's an example where organ imaging or blood-brain barrier permeabilities. Here's some examples of the data. And of course, we have application notes to address the mix and match and flexibility of Sapphire FL. Like one example here is a mega stock dice, which has not fulfilling or not able to image under our standard, let's say, fluorescent channels. You can see here the excitation emissions are totally different. Okay, right. So we can see here that the very huge stock shift excitation emissions. Okay. And of course, also VC assays for in vitro protein ligand interactions, plate-based screening or visualizations, and also in vivo imaging and monitoring tumors in the mice. We have a lot more in our application notes on our website, or please feel free to look it up on our website or contact our local distributors if you're interested in some of these applications. In a summary, a Sapphire FL is the ultimate imager for flexibility. Okay, so although it is not a microscope, okay, and it is not a microscope, it's not really a dedicated in vivo imaging equipment, okay, like for 3D imaging of your mice, but it is a very good intermediate, or I'll say a very depending on your research, it can be a very good intermediate between your normal gel documentation to confocal microscopy, or between your documentation to 3D screening, 3D imaging of your mouse, or can be something like your generic upgrades for your fluorescence screening other than your gel documentations. Okay, so it offers a very huge specs over here to ensure the best signal to noise ratio and dynamic range. The uncoupled optical module design, now right now we have 11 standard optical modules available for you to use. Okay, this is how the system looks like. Okay, with a built-in gaseous anesthesia port. So it's like a box size, not real box, but it's shaped like a box. We've opened up with a lid, and here's the imaging stage where you put your sample on top of the glass stage. Okay, here's some uh, samples we can see. It supports in vitro to in vivo studies, and we have these representative installations all around the world right now. NIH, like for Hong Kong Creamery University, Hong Kong U universities, and also for like Griffith University in Australia. We have some cancer and also nuclear research for the phosphor imaging, and recently for in Korea. We have installation in Gyeongsang University as well. We have, I think, a few. Okay. And then, of course, we're talking about Sapphire FL. We have a lot more installation on the 2017 version in Korea as well. So Azuba system is a Western blot solution provider. So we have imaging systems for you to capture images of these, your Western blot, either in chemi, luminescence, or fluorescence signals. We also have gel system, reagent, consumables for your Western blotting needs, okay, except for antibody, okay, primary antibody, right? So we have imaging solution for your lab and applications. These are all the equipments we have in our company portfolios. And I am Kylie. Thank you for your attention. Okay, and thank you, Philip Korea, for this organization.